Hello, and welcome to Improving Sample Prep Efficiency and Data Quality of Challenging DNA Samples, brought to you by Forensic and sponsored by Thermo Fisher Scientific. My name is Michelle Taylor, Editor-in-Chief of Forensic, and I will be your moderator throughout. For today's webinar, you will earn 1 hour of continuing education credit. Following the conclusion of the webinar, you will receive an email with information on how to obtain CE credit documentation. We have a great lineup scheduled to present to you today, but before we begin, I'd like to take just a moment to cover a few logistics. At the end of the presentations, we will hold a question and answer session. To ask a question, click on the Ask a Question tab in the upper right corner of the screen. Please also take note that the right side of the screen features an overview of today's webinar, as well as more information about our speakers. If you have a technical question during today's event, click on the Test Your Connection button at the bottom of the screen. From there, you can access additional webinar support. We also invite you to use the social media widgets beneath the webinar to share with your friends and colleagues. Today, you will hear from Dr. Sheree Hughes, Associate Professor of Forensic Science at Sam Houston State University in Texas, as well as a few of her graduate students. Dr. Hughes completed a PhD in Health Sciences/Forensic Genetics at Bond University on the Gold Coast in Australia, investigating forensic DNA typing methods for highly degraded samples, such as those recovered from mass disasters, missing persons, and historical cases, in conjunction with DNA repair techniques and phenotypic SNP analysis. Natalia Czado is currently a doctoral student at Sam Houston State University. She earned her master's in forensic science in 2015 and completed her bachelor's degrees in biological sciences and criminology at North Carolina State University in 2013. She was a lecturer in forensic science at Fayetteville State University from 2015 to 2020. Natalia's research interests include DNA profiling of challenging and degraded samples, and she has authored/co-authored several papers in this area. She is hoping to branch out into genetic genealogy and has recently completed Boston University's Genealogical Principles course. You'll also hear from doctoral student Kayli Carrillo. Kayli's research interests include optimizing DNA collection, extraction, and enhancement methods for low template samples. Prior to attending Sam Houston, Kayli received a Bachelor of Science in Biology from the University of North Texas. The last doctoral student you will hear from today is Jennifer Snedeker. She is interested in advancing forensic DNA and human identification methods by optimizing DNA extraction procedures from skeletal remains and examining novel investigative methods for these sample types. Her research is focused on highly degraded and challenging skeletal samples that closely mimic those seen in forensic casework. Prior to beginning her PhD research, Jennifer received a Bachelor of Science in Human Biology from Michigan State University. Thank you for joining us for today's webinar, and keep your eyes open for an invitation to our next one. Now, without further ado, I'll hand it over to Dr. Hughes to get us started.
Thank you very much for the opportunity to speak today. So first of all, our quick disclaimer is to say that the data and the opinions that we are going to present today are that of our own and not of Thermo Fisher Scientific. Also, I'd like to disclaim that a lot of the instrumentation and kits were provided by Thermo Fisher today. However, there was no financial remuneration at all. And last but not least was also because of some of the sensitive nature and imagery in this presentation, we also request that there is no screenshots or pictures taken of any of those images throughout this presentation. Thank you. So of course, this work would not be possible without my fantastic team, and I am 3 of my PhD students that I have presenting today on each of their representative areas, Natalia, Kayli, and Jennifer, and you'll hear from those throughout this presentation. So overall, the objective of the work that we are presenting today was really looking at how we can examine the utility of the HID NIMBUS Presto system for its effective and efficient DNA purification from really difficult and challenging sample types that you may find in a lot of casework. So these will include fired and unfired cartridge casings and DNA recovered from those touched items. Also, other touched items that may be commonly retrieved from for casework and crime scenes, such as water bottles and computers and phones, etc. And then also a bit of a plethora of challenging samples from decomposing cadavers. So we're going to look at recovery from hair, nails, and teeth, as well as some bones from skeletons that have been environmentally challenged in different ways. So a little bit about the Nimbus Presto system. So starting with the Kingfisher Presto purification system, and this is working on an automated magnetic particle base purification system. As you can see working behind you there, it's really efficient and effective, and it's quite a swanky little thing that works well. It actually has a really neat magnetic rod system that has these tip combs that work for the purification, and this nifty little turntable system that allows for sample handling and being able to load your samples and move them around the deck. And of course, that system is then coupled with a liquid handling system. And the way we've done it in our lab is with the Hamilton Microlab Nimbus HD. And it can process, as you expect, up to 96 samples, and does that in a very, very effective way in combination with the Kingfisher Presto. And it's extremely adaptable. It's an adaptable system into whatever platforms that you would like and the formats that work for you. And we've found it to be working very seamless and efficient in our lab once it was up and running. And the scripts that we have on this system that we're going to talk about today are able to use both the PrepFiler and the PrepFiler BTA chemistry for our sample types. And again, these samples are lysed manually before we put them on the machine. So we have the lysis step performed first, and then we put it onto the system for all of the purification after that. So we really does allow for this high throughput capability as you would want in a crime working lab, or even in a very high throughput research lab. And it's a very flexible format. You can do anywhere from 4 to 8 samples all the way up to 96 on a deck without very little time difference. So it does reduce the analyst's hands-on time quite significantly. And if you're looking at batches of, say, 12 samples is what you may be looking at, similar to automated, say, on the Automate Express. It's essentially 40 minutes versus almost 2 hours if you're doing it even manually by hand, which we will be drawing a couple of comparisons and conclusions on throughout this presentation. Another really handy tool that we find is extremely useful in the lab, particularly even just starting with a lot of students getting familiar with the instrumentation, the software, how to have it working properly. The GUI system really does make this a piece of cake. It's really quick and easy to follow. It's quite intuitive. It enables you to custom the adaptability of what you want to use for your user preferences in terms of plastics and workwear. It eliminates the risk of a lot of setup error, as well as obviously handling contamination. Being able to set up with this GUI system allows you to make sure that you've got all the chemistry, all the consumables and all the plastics all in the right place, doing the right thing. It has a sample and reagent tracking capabilities if that's something that's really important for your lab and working in with your LIMS system, etc. And also a lot of guidance for setup. So in essence, it prevents anything from going wrong. So we found that extremely useful and very, very advantageous when it comes to the liquid handling side of things. And as you can see, you can just choose, it allows you to walk you through the deck setup. So it's very, very easy. It's got this swanky little color system, and it allows you to set up the deck efficiently, effectively, and correctly every time. So the two chemistries that we are talking about and that we're looking at is our PrepFiler BTA for all of our bone samples and some of our other casing samples as well, and also PrepFiler for our stock standard, your blood and swabs, etc. So both of these chemistries for these various types of samples, with the scripts are able to very effectively work your solutions from start to finish on the Kingfisher Presto and Nimbus systems. So for DNA quantification, after our extracts with our elutions run through our quantifiler trio quantification kit, and most of you will be familiar with all of the targets that are used. We have our 2 human targets, our small and large, which of course allows for the degradation index, to be able to give you some sort of indication of your DNA quality. So DIs 1 to 10 is indicative of some slight to moderate degradation, whereas our DI values greater than 10 would it definitely tells you that there's some significant degradation there. And sometimes you would get this undetermined, where you're unable to determine your DI. Well, of course, that's extreme degradation and or potentially some significant inhibition there as well. But in terms of other targets, you also have your male target, of course, and your IPC, which allows you to obviously detect PCR inhibition with when your delta IPC from your controls, of course, is greater than 1 cycle. In terms of PCR implications, so our STR kit we're using for this work is the Verifiler Plus PCR implication system. And this is a great system for really low template and challenging samples, which we are going to cover today, is the ability to increase your input volume, so up to 17.5 microliters. So this really does allow some increased sensitivity and being able to put as much of that really low template extract into PCR, into the PCR reaction. We have our 23 autosomal markers, as you would expect in these new kits. We have a Y indel and, of course, a melogenin. And also, we have our now 2 internal quality control markers. We have one short at around 70 base pairs and then the longer at 451. And, of course, these are used to help basically confirm the success of your PCR and also confirm some inhibition and degradation within your sample. So as a quick reminder of how these internal quality control markers work, this is essentially what they look like. We've got a nice, beautiful sample here. Our control markers are nice and balanced. All of our peaks look good. We've got a really nice sample. When we have a sample with some significant amount of PCR inhibitors involved, you can see that we have now our large quality control marker has now dropped out. So this will be either very small or dropped out, and confirming that we've got some significant inhibition here, as opposed to degradation, where our markers are nice and reasonably balanced. And however, we do see that significant ski slope and decrease in our peak heights, particularly, and drop out of our larger markers, as you'd expect. So of course, these markers are extremely useful for being able to get a snapshot and confirmation of the quality of your sample and your PCR reaction, how it's worked, and of course, then your STR profile in front of you. In terms of our analysis, here are the students in their room hard at it. So of course, with the Verifiler Plus, just very quickly, we targeted a 500 picogram DNA amount for our PCR, and only throughout all of these studies, only samples that yielded some detectable DNA, so greater 1 or greater than 1 picogram per microliter, were continued on for STR typing. All of these samples were run and were separated and detected with the fragment analysis on 3500, and our data analysis was performed with GeneMapper IDX version 1.6, with our analytical threshold being 100 RFUs and our ST being 700 RFUs. So overall, the workflow for this study that we're presenting today is looking at a combination of difficult samples. So here are our brass casings that were collected with using various methods, which Natalia is going to talk about. We then have some swab touch items and some challenging samples from decomposed cadavers that Kayli is going to discuss. And then lastly, with our skeletal remains, we have some buried surface burial, some different types of bones, etc., from different environmental insults that Jennifer is going to talk about. All of these samples were then DNA extraction and purification was performed on the Nimbus Presto, HID Nimbus Presto, and then run through DNA quantification with quantifier trio, as I explained, and also STR typing was performed with Verifiler Plus. We also ran some comparisons by performing all of these samples were ran concurrently with using PrepFiler or PrepFiler BTA as determined by sample type, and these were performed manually as well. So we've got comparison of on the automated platform versus manually performed in the lab, and we're going to present those results too. So I'm now going to hand over to Natalia to run you through all of the results from the brass casings. Thanks, Natalia. Over to you.
Thank you, Dr. Hughes. As many of you know, firearms and ammunition are some of the most common types of evidence submitted to crime laboratories. DNA can be deposited on the surfaces of guns and cartridges during the handling and firing of a weapon. Using this DNA to generate profiles can help investigators identify suspects and persons of interest in violent crimes. However, obtaining DNA profiles from these samples has historically been difficult. The difficulties stem from several factors. First, these types of samples normally have low amounts of DNA deposited on the surfaces. They are most often thought of as touch or low template samples. In addition to this, any DNA present could exist as a mixture, further complicating DNA profile interpretation. Lastly, the metal composition of the weapons and ammo plays a role in DNA recovery and genotyping. Metal ions can bind at several different sites on the DNA template. These strong bonds form metal ion complexes that can make DNA recovery from the metal surface more difficult. So not only do analysts have to consider how best to recover low amounts of template, they must also consider whether their method will adequately release the DNA from the surface. Also, these complexes can break one or both strands of the DNA template, depending on the binding site. Although many types of metals interact with DNA, obtaining DNA profiles from ammunition with copper or brass surfaces is most challenging. This is due to copper's ability to bind at more sites on the DNA and produce reactive oxygen species that lead to oxidative damage of the DNA template. Damage and degradation of the DNA is detrimental to our size-based detection methods, and the presence of metal ions in PCR can inhibit the polymerase. So to summarize this, we have an issue collecting enough DNA. The DNA we collect is often fragmented and bound to ions, and the ions are inhibiting the amplification of the low amounts of good quality DNA template. Not really a recipe for success. For these reasons, there has been a lot of research over the years in how best to collect low template DNA, especially from the surface of ammunition. One of the most popular DNA collection methods is double swabbing, where the first swab is moistened with sterile water and a subsequent dry swab is used to collect any remaining genetic material. Although this method is inexpensive and works for a variety of surfaces, there has been mixed success reported for collecting DNA from ammunition. To improve DNA recovery, soaking has been tested. There have been several different protocols proposed. The one developed by San Diego PD's Forensic Biology Lab is probably the most well-known. However, increased success was only minimal using this collection method. Both swabbing and soaking prior to DNA lysis and purification do have the inherent risk of losing precious amounts of DNA. Therefore, direct PCR has been examined. By bypassing extraction and adding the swab directly into a PCR reaction, about 60 to 70% more DNA could be amplified. Although direct PCR works great for other low template samples, success has been inconsistent for DNA recovered from metallic surfaces and ammunition. And this is probably due to the continued presence of metal ions in PCR without the purification step. Because of the impact that metal ions are thought to have on downstream genotyping, the use of metal ion chelators has been examined. One study examined moistening a swab with 0.5 molar EDTA rather than sterile water. The researchers reported that less oxidation took place and enough good quality DNA template was recovered to successfully sequence mitochondrial DNA. More recently, a rinse swab protocol using a solution comprising bovine serum albumin and glycine-glycine histidine tripeptides, both great metal ion chelators, has been proposed. The solution is referred to as BT mix and has been moderately more successful than previous collection methods, but is time-consuming and does not consistently generate complete STR profiles. Currently, no method has consistently recovered enough good quality DNA to generate full profiles. Collecting the small amounts of DNA deposited is the most important consideration for these types of samples. Therefore, more research in determining a more effective collection method is warranted. In a recent study completed at Sam Houston State University, we determined that automated extraction methods based on magnetic beads could adequately remove inhibitory metal ions. Using PrepFiler BTA chemistry, we could still obtain full DNA profiles in the presence of 0.1 molar copper and 0.03 molar zinc, which are concentrations much higher than what would be expected from the surface of ammunition. Because of this, we wanted to examine whether including PrepFiler chemistry during the DNA collection stage and pairing it with extraction on the HID NIMBUS Presto system could increase both the amount of DNA we collect and the number of samples we can process in a short period of time. For this portion of the study, we sterilized 36 cartridges. Through some miracle, we were able to get enough rounds of the same type of ammunition in the middle of an ammunition shortage. We selected a brand of brass cartridges since these are the most problematic for DNA profiling. We made buccal cell suspensions from a single donor and spotted the equivalent of 10 nanograms of DNA onto a marked location on the cartridge. All of our samples were fired by a male law enforcement officer using a Glock 19. We took 12 of the 36 casings and used them to test the PrepFiler lysis buffers as moistening agents. So 6 were moistened with PrepFiler BTA lysis buffer and continued on to the BTA extraction protocol on the HID NIMBUS Presto system. The other 6 underwent the same process using the standard PrepFiler lysis buffer. The other 24 casings went on to comparison testing. 12 were moistened with water and then divided between PrepFiler and PrepFiler BTA extraction on the Nimbus. The other 12 underwent the BT mix rinse swab protocol, followed by each type of PrepFiler extraction. Because the BT mix protocol requires the use of 2 swabs, they had to be placed into separate extraction tubes. And after extraction on the Nimbus, eluates from the same casings were pooled and concentrated to a final volume of 50 microliters. All samples were then quantified with QuantiFiler Trio with the addition of BSA. I want to mention that a similar workflow was repeated for the samples that underwent manual extraction. We compared the DNA quant data for all of the automated samples, examining DNA yield, inhibition, and degradation. We did not observe any inhibition, but all samples exhibited some level of degradation. DNA was only recovered for 22 of the 36 samples, meaning that 14 of the casings resulted in 0 or undetermined amounts of DNA, which is not unexpected for fired brass casings. Extremely low amounts of DNA were recovered, with 134 picograms being the highest total amount recovered. The combination of BT mix rinse swab collection paired with PrepFiler extraction was the most successful. When we compared manual extraction to automated, there was no trend observed for DNA recovery. For most samples, automated extraction was able to recover more amplifiable DNA. However, any differences were not statistically significant. We then examined genotyping success. Again, only samples that recovered at least 1 picogram per microliter of DNA were amplified. For casings, this means we only had 3 samples for both manual and automated extraction move forward to DNA amplification. None of the samples generated a complete profile, with allele recovery ranging between 2 and 17% for our automated samples. Although samples for manual extraction were amplified, all 3 exhibited a mixture of an unknown contributor. We have not yet completed mixture analysis, so these results are not listed here. I think it's important to note that none of the automated samples exhibited mixtures. This example highlights that processing samples in an automated workflow decreases the risk of contamination. So we recommend using an automated platform for DNA extraction from low template samples. That wraps up the results for the casings portion of this study. So now I'll be handing it over to Kayli to discuss the next section.
Thank you, Natalia. As mentioned, the next sample type we will be discussing is touched items. Touched DNA is often referred to DNA that is transferred to an object or another person during physical contact, which is often encountered during property-related crimes. Touched items present their own unique set of challenges due to the limited amounts of DNA present. With low amounts of DNA, there can be stochastic effects such as allelic peak height imbalance or allelic or locus drop in or drop out that can further complicate interpretation. As you can see in the photo, touched DNA is typically composed of cell-free DNA, endogenous or exogenous nucleated cells, fragment-associated residual DNA, which is the result of apoptosis or cell death, and anucleate corneocytes. The outermost layer of your skin consists of keratinocytes, also known as corneocytes, that have undergone keratinization. So it is thought that corneocytes do not have a lot of nuclear DNA, and therefore do not constitute a significant source for touched DNA. However, DNA deposited on touched items is highly variable and not easy to predict due to the wide variety of factors that can affect touch DNA yield, such as the skin shedding status of an individual. An individual could have sweaty or dry hands, or the frequency of hand washing can all lead into this idea of a good versus bad shedder. Another factor to take into account is the type of contact. Does this person use a little or a lot of pressure when touching the item? Also, the substrate surface can affect DNA yields, as DNA yield tends to be greater with porous or rough surfaces. All these combined factors impact DNA transfer, persistence, prevalence, and recovery, or TPPR. In order to help recover as much as possible from these touch samples, there has been an improvement on the increased sensitivity of PCR kits, such as GlobalFiler IQC and Verifiler Plus. In this project, 2 swabs were moistened with DI water and were used to sample cell phones, keyboards, and bottles from 6 individuals. 1 swab was processed using the HID NIMBUS Presto, and the other swab underwent manual extraction. Both methods followed the PrepFiler kit protocol. Now for our results, we can see that most DNA was recovered from our keyboards and the lowest from our cell phones. But all of our samples had at least 0.0067 nanograms per microliter amounts of DNA recovered. And degradation was not an issue for our samples, as we did not see a ski slope effect in our STR profiles, and our degradation index only ranged from 1.36 to 3.46. Now, if we take a closer look, we can see 13 out of 18 samples indicated mixtures. As you can see in this electropherogram of a cell phone, of the clear appearance of mixtures. And mixtures in our samples is not unexpected, because just think of how many times you either shake hands with a coworker or touch a doorknob, and just how much you touch your cell phone in general, and all that transfer of DNA that is possible. Even though there were mixtures, we did see 100% allele recovery from the major contributor or the owner of the item. So right now we are currently running through a mixture interpretation software for the minor contributor. Now when we compare the HID NIMBUS Presto and manual extraction, we can see that the touched samples extracted with the HID NIMBUS Presto generated extracts with significantly higher DNA yields. Next, we will be discussing challenging samples from decomposed cadavers. For this portion of the project, 2 samples were collected from each cadaver, 2 hair, nails, and teeth, from 6 cadavers that were placed on the surface for up to 3 months. But these challenging samples come with its own unique set of challenges. While hair is a commonly encountered evidence type, it may be limited in its DNA quantity and quality, especially if the root is not present. The keratin that is present in both hair and nails is known for degrading and inhibiting DNA as well. And for teeth, while it is more resistant to degradation, it is still subject to environmental contamination that can negatively impact DNA yield. Additionally, the calcium and enamel present in teeth can also act as PCR inhibitors. To prepare these samples, hair was cut 3 to 5 millimeters, and if the root was present, was included. Hair was washed with a turgidzyme detergent in order to wash away any exogenous DNA. Then with water and following with ethanol. Hair was extracted using the PrepFiler protocol. Our nail samples were not simply just nail clippings, but the entire nail bed itself, from either the thumbs or the big toe. We received both a left and right nail for each cadaver. And as you can see in the photo, we cut each nail in half, and from that half, a 5 millimeter cut was made so that we could proceed with the HID NIMBUS Presto workflow or manual extraction. Similarly to hair, nails were extracted using the PrepFiler protocol. Finally, for our teeth samples, premolars and molars were washed with 10% bleach for 3 seconds, then water and ethanol. Teeth were wrapped in a Kim wipe and pulverized with a mallet before being powdered with a freezer mill. Teeth samples were then extracted using the PrEP Filer BTA protocol for both manual and automated extraction. Now, if we look at our results for the challenging samples, our highest amount of DNA was recovered from our nails. This can be explained because, like I previously mentioned, we were using our nail beds and not necessarily just nail clippings. Nail beds have adhering soft tissue, which can explain the higher amounts of DNA available. But in our samples, there was no inhibition detected, but DNA degradation was observed in our nail and teeth samples. And while we know that these are challenging samples, we were still able to achieve over 80% allele recovery from our hair, nails, and teeth. And here is a picture of an electropherogram of one of our nail samples with 100% allele recovery. And we can see that there are good peak heights and a balanced profile throughout this sample. When we look at the hair samples extracted with the HID Nimbus Presto versus manual extraction, the HID Nimbus Presto generated statistically significantly higher DNA yield in comparison to manual extraction. Additionally, more hair samples met our 1 picogram per microliter threshold to proceed with STR analysis. Similarly, for nails, the HID Nimbus Presto also yielded significantly more DNA yields than manual extraction, and also yielded more samples that met our threshold. For our teeth samples, there was no statistically significant difference between DNA yields between the HID Nimbus Presto and manual extraction. All of our manual extracts and 10 out of 12 samples using the HID Nimbus Presto met this threshold. But in summary, you can see that the HID Nimbus Presto either performed similarly or significantly better than manual extraction. Now, on to Jen, who will discuss the much anticipated bone results.
Thank you, Kayli. As she mentioned, the last sample type we'll be discussing throughout this webinar are our skeletal samples. These samples are very important because they are often the only remaining piece of evidence in several situations, including cold cases, missing persons, or mass disaster victim identification. These samples are unique in that the reason they are often the last source of evidence is the same reason they can be so challenging to process. The inherent nature of these samples allows for that DNA to be protected within the tough hydroxyapatite matrix of the bone. But this requires a very complicated sample preparation process to actually access that DNA. Not only that, but these samples are also often highly inhibited from either endogenous sources like calcium or collagen, or exogenous contaminants like humic acid found in the soil. Additionally, these samples are usually highly degraded and naturally contain low levels of DNA. Therefore, we wanted to investigate the success of the HID Nimbus Presto system for DNA extraction from these challenging sample types. We collected our samples from STAFS, the Southeast Texas Applied Forensic Science Facility, which is a willed body donor program at Sam Houston State University. Individuals or family members can choose to donate their bodies or their loved ones to this facility for research or education purposes. I'm lucky enough to work out at STAFS and to utilize it for my research. We're very thankful for our donors, and this is what I would like to remind you all to please refrain from taking any photos of the slides that contain cadaver images. With that being said, this project we collected from 3 different insult categories: surface decomposed remains, burned remains, and buried remains. 6 cadavers were collected for each insult group, and 5 skeletal elements or bones were collected from each cadaver. Like I mentioned previously, the sample preparation for skeletal samples can be very tedious and time-consuming, but it is doable. First, a window cut needs to be taken from our larger skeletal elements, like this femur you see here. This is done using a Dremel power tool. Smaller samples skip this window cut step, and instead the entire sample is processed as a whole. Here you see this window cut taken away. And once we have that window cut, or the entire sample that will be processed, we sand down the outside surface in order to remove any exogenous DNA to limit contamination moving forward. This is again done using a Dremel power tool, but with a sanding bit attachment. At this point, we're ready to chip our bone into smaller pieces to make our powdering process easier. And these chips go through a series of wash steps to again limit contamination. And at this point, we're ready to begin our powdering. At SAM, we use a freezer mill, and this works by pouring our chips into the vial you see on the left. In that vial, there's a magnetic rod. So you place this vial in the freezer mill, which is located in the center image, and this freezer mill is filled with liquid nitrogen. When we turn it on, that magnetic rod is going to move back and forth very, very quickly, pulverizing the chips into a fine powder like you see here. And at this point, we're ready to begin our extraction process. You can see there's a lot of hands-on time that occurs up front during the sample preparation process. And that's why we were interested in looking at the HID NIMBUS Presto system for DNA extraction from these samples to limit hands-on time at a later step. The first insult type we'll be discussing for this project are our surface decomposed remains. At staffs, upon intake, the cadavers are placed on the surface under cages for extended periods of time. The samples collected for this project were on the surface from anywhere between half a year to about 2 years. While in the field, these cadavers are exposed to a variety of environmental insults, including sun bleaching or UV damage from that hot Texas sun. Additionally, these samples can interact with the soil, leading to potential inhibition, as well as interactions with different microbial communities or even scavengers if they're able to make it under the cages. They're also exposed to moisture from rain or humidity, and they have that extreme Texas heat impacting them. So anything that the natural environment goes through, these samples go through, which is going to lead to a very challenging sample type. For this project, we collected 3 long bones, the humerus, femur, and tibia, and 2 foot bones or tarsals, the medial cuneiform and the first metatarsal. For each cadaver, we collected either the left or the right side, but not both. Our burn samples are our next type of insult, and these were processed by individually burning each element separately, as you see in that top image here. So the skeletal elements were placed on a makeshift grill of wood, charcoal, and chicken wire to hold the bone, and the bones were burned until they reached a black or brown coloring. At this point, we see that there's DNA damage and degradation induced on these samples. For our burn samples, we again collected our 3 long bones, the humerus, femur, and tibia, and then 2 other foot bones, the cuboid, and then the first metatarsal. The last insult category is our buried samples. At staffs, cadavers are buried at various depths and for extended periods of time based on the research study. For this particular study, the cadavers were buried for a length of between just a few months to almost 4 years. We often find that these are our most challenging sample types as they are highly degraded and inhibited, likely due to the acidity of our soil from our pine trees, as we are part of the piney woods of Texas. Additionally, these samples often have adipocere formation, which is commonly seen in buried samples where there is a lot of moisture. This is when the decomposing soft tissue forms a waxy, fatty substance that can further complicate the DNA extraction process from these sample types. For our buried samples, we collected the humerus, femur, tibia, medial cuneiform, and first metatarsal. So overall for this project, a total of 30 samples were collected for each insult type and extracted in duplicate on the HID NIMBUS Presto system using the PretFiler BTA chemistry. Here we are looking at the average DNA yield per insult, and you can see that the burn samples had the largest variation, with some having relatively high yields. But on the other hand, our buried samples had a limited spread with a much lower recovery. From the 180 extracts, 133 of these proceeded forward for STR typing, meaning they met that 1 picogram per microliter input value. 57 of these were surface samples, 44 burned, and 32 buried. When we compare our allele recovery versus DNA input, as expected, the more DNA we're able to target, the higher allele recovery we see. For our surface and burn samples here, we see that if we could target at least 0.2 nanograms of sample, we have an 80% allele recovery or greater. The only exception is that burn sample that you see has 0.5 input value, but about allele recovery of 63%. And I want you to keep this sample in mind when I move to the next slide. When we look at our buried samples, we see that this trend isn't very consistent, and we have a much lower allele recovery regardless of our DNA input. And much of this can be explained if we examine the degradation index of these samples. So the majority of our surface bones you see here have a DI between 1 and 5, which means they're slightly degraded. And a few samples pass that threshold of 10 DI, meaning that they are severely degraded. And that same trend is consistent with our burn samples, with the majority between 1 and 5, and we see one sample has a DI around 15. So this is actually that sample I mentioned on the previous slide that had an input of 0.5 nanograms, but a low allele recovery. And that's due to this high degradation index and the dropout of those larger loci. So this highlights some of the challenges we see when processing these sample types. We have to look at the sample as a whole picture. So we have to look at the amount of template DNA, if these samples are degraded, and then furthermore, we have to look at our inhibition of these samples. And all of this will predict our success in allele recovery. When we look at our burnt or our buried samples, we see that we have much higher degradation indices, indicating those lower allele recoveries. And this is not unexpected as this is a common issue we face with these challenging sample types at STAPS. So from here, we're going to compare our HID Nimbus Presto extractions to our manual extractions using the PrepFiler BTA chemistry. And in this figure here, we're looking at our quant data. And we see that overall, there was no significant difference in DNA yield for our surface or burn samples. But when we compare our buried samples, we found that the samples processed using the HID Nimbus Presto system had a statistically significant increase in DNA yield. However, regardless of the method used, these values were still very, very low. When we compare which samples meet that 1 picogram per microliter threshold for PCR amplification, we see that the Nimbus had more surface samples and buried samples proceed forward. This demonstrates that even if the DNA yield was not significantly better with the Nimbus, it resulted in a higher chance of success in continuing through the HID pipeline. Unfortunately, at this time, we are still working to complete our STR typing for these manual extractions, so we are unable to compare our allele recovery data. However, we were able to process a subset of these samples using other bone extraction methods that we commonly use in our lab. This includes the Automate Express, which utilizes the PrepFiler BTA chemistry, and our lab's Total Demon method, which is an adaptation of the Loré method. And when we compare these 4 methods, we found that we had no statistically significant difference in DNA yield, regardless of which extraction method was used. This means that the HID Nimbus Presto system performed on par with the other DNA extraction methods we implement in our lab. But outside of DNA recovery, there are some other variables that should be considered when choosing which DNA extraction method to use for our skeletal samples. For instance, using the HID Nimbus Presto system allows for 96 samples to be processed at once, compared to the only 13 that can be processed on the Automate Express or comfortably processed manually. Furthermore, utilizing the PrepFiler BTA chemistry, whether that be on the Nimbus Presto, the Automate Express, or manually, only requires 50 milligrams of bone powder versus the 250 milligrams used for our Total Demon method. And this is saving our precious sample type. And lastly, utilizing the partial demineralization chemistry of the PrepFiler BTA kit saves time, reducing the extraction process to hours rather than days. And if the HID Nimbus Presto is used, the hands-on time can be further reduced, as Dr. Hughes is about to summarize.
Thank you, Jen. So now I'd like to draw your attention to really the workflow comparison altogether. So if we start with the PrepFiler BTA system, whether we perform that manually or on the Automate Express or the HID Nimbus Presto, if we look at a batch of, say, 12 samples to compare, it's a great place to start. So with a set of 12 samples manually, there is really no difference in the beginning steps. So from sample prep, going from bone chips down to the bone powder, it's exactly the same. Going through to the digestion or cell lysis step, it's the same. Now moving that onto extraction and purification, performing that manually with 12 samples, that step is just shy of 2 hours. So when you look at PrepFiler BTA solution chemistry being performed manually, it's a total time of around about 16 hours and just over 2.5 hours of hands-on time. When you compare that to the Automate Express and the HID Nimbus Presto work solutions with those 12 samples, there's a vast difference. Even though in totality is about 14 hours, it's really the hands-on time down to less than half an hour or so, really does allow the analyst to, free up their time to do other things. And even though the 12 samples on the Automate Express and the HID Nimbus Presto is comparable in terms of time and hands-on time, that's with 12 samples. The real neat thing with the HID Nimbus Presto, of course, being able to increase the throughput up to those 96 samples, the increase in time on the instrument is actually quite negligible. Another 10, 15 minutes, and you've gone from 12 to 96 samples. So that is the real advantage here using this combined Kingfisher Presto and the HID Nimbus system as a whole. The same holds true with prep phylochemistry, slightly different. We have our, of course, moving through from cell lysis is the same again, down through the automated platform extraction and purification. Those differences you can see range from everything from an hour and a half performing it manually down to about half an hour, 40 minutes on the automated systems. And again, exactly the same advantages. Over 2 hours or so for the hands-on time with the prep filer kits, you performing it manually versus your half an hour on the Automate Express and your HID Nimbus Presto. But of course, remembering that you can increase that throughput very, very easily with negligible time. So it's a really fantastic solution for all of your DNA extraction and purification samples in the lab. So in summary, the HID Nimbus Presto system really did work really well in our laboratory with these types of really challenging samples. They performed as we would expect with such difficult samples. They were able to purify DNA from a variety of all of the different difficult samples that we encounter in our lab all the time. When we compared DNA yield, the Nimbus Presto really did perform as we would expect to see. And very similarly, if not better, with manual extraction methods that we use in our lab for those different types of samples. And overall, the big advantage is the HID Nimbus Presto system really does save a lot of analyst time by limiting those hands-on requirements and allowing for that 96 sample throughput all in one time, very, very effectively and efficiently. So that is it from us. I'd like to, of course, acknowledge a few very important people. This work wouldn't have been possible without the immense support from the Thermo Fisher team that work with us, namely Yvonne, Angie, Han, Laura, of course, Andrew and Patrick on this project. Of course, I have to acknowledge Southeast Texas Applied Forensic Science Facility, or STAFS, for providing us with some samples from their donors. I need to thank their donors and their loved ones, because of course, without them, this type of research is just not possible, and work is it's very, very difficult to do without these types of real samples. And of course, the Department of Forensic Science and the Institute of Forensic Research, Training and Innovation for their support at Sam Houston State University. So if you have any questions, please feel free to reach out to any of my 3 PhD students here in their respective areas, or myself if you have any other generic questions or general questions on this type of research that we perform here at Sam Houston State with all of our very, very difficult and challenging samples. Otherwise, if you have any instrumentation questions, please feel free to reach out to Han. His email is there, or your local representative, I'm sure will be able to help you with anything. Thank you so much. I'd like to hand back to Michelle now, and I think she's going to moderate the Q&A session. Thank you.
Thank you, Sheree and panelists, for those interesting and informative presentations. We do have time for our Q&A, so if you have a question for the panelists, please submit it through the Q&A panel on your screen. I also want to draw your attention to the Resources tab on the right side of the screen. There are a few resources there connected to today's webinar that you'll probably find really interesting. While we wait for everyone to submit their questions, I have a couple polling questions for you. Our first polling question is, does your lab use automation? Answers are A through F, and please check all that apply. All right, let's see. Most of you do not use automation, but a little less than half are. Let's go to the next one. Which areas are your greatest need for automation? And again, please feel free to check all that apply, A through E. Wow, everyone says all of the above, 100% on that. Last question before we get to the Q&A. Would you like to receive more information about the Nimbus Presto system? All right, thank you, audience, so much for participating in our polling questions. Now we get on to the extra fun part, the Q&A. Kayli, our first question is for you. Are you ready for us?
Yes, ma'am.
All right. So are there major differences between bone and teeth prep?
I would say that the main difference between like bone and teeth prep is mainly like the size of the sample. So with bone, you know, you have a larger sample usually, but with teeth, we use, as we saw in the presentation, usually a mallet to pulverize the bone. So that would save a lot of time. You just kind of smash the tooth instead of using maybe like a Dremel that we do with bones. But you still do the same protocols of washing it and powdering it. So I would say just that sample prep is the main difference.
Gotcha, okay. Natalia, you're up next.
All right.
How long can the swabs from cartridges be stored and still have success in isolating DNA? And how do you store those cartridges?
Okay, so those are all fantastic questions, and there are a bunch of different variables we have to consider when answering that question. So as many of you know, the longer that DNA is exposed to the surface of copper, the more detrimental those effects are going to be on the DNA. And so I would definitely recommend that if your lab does use standard swabbing as the collection method of choice, that you don't just leave the casing stored, but you do go ahead and swab and then store the swabs. Now, we haven't measured or tested storage time here at SHSU, and a lot of that really does depend on the collection method. So with standard swabbing, storage is definitely an option. However, if your lab is going to adopt the BT Mix rinse swab protocol, you can't really store the swabs for that. And the reason is because you are using that rinse solution in the same tube as your swab. So it all gets collected together. And so if that is your method of collection, then I would actually recommend that you go straight through extraction and then store the extract. And that way you're preventing any more damage and degradation from those copper ions. And you're at a place where you're not storing potential ions in that rinse solution as well. Hopefully that answered the question.
Absolutely. Thank you so much. All right, Jennifer, let's get you into the mix. We have a question for you.
Sounds good.
How long does sample preparation for bones take?
So it's really dependent on the method and sample type being used for each lab. So for instance, for our lab, we're processing that entire window cut because we want to have a large pool of powder to sample from. But other labs that maybe are just trying to target enough powder to actually, worth that sample might choose to only process a few chips at a time. So that could change the sampling process time. But of course, every sample is going to require washing because we want to make sure we're removing any contaminants. And once we wash, we're of course going to have to dry. We opt to dry overnight just because the timing of it gives you a little more variability. But you can also choose to use an oven, which will speed things up. But of course, there still is that wait time for that. So pretty much from beginning to end for each sample, you're going to take roughly a day or a day and a night. So it is a time-consuming process, and it's one that is very tedious, but if you do it right, it helps things out quite a bit.
Gotcha. Okay, thank you, Jennifer. Dr. Hughes, this is a question for you or pretty much anyone else, but maybe we'll start with you since we haven't heard from you in a bit. How difficult or time-consuming was it to set up the instrument, add reagents, tips and plates, and what about running the software, et cetera, that whole kind of process?
Yeah, well, setting it up, obviously, and everyone getting familiar with the way it all works and the scripts running smoothly, obviously is what took the longest time, but we had immense support from the Thermo Fisher team to be able to make sure we had everything up and running. But of course, the GUI system, the GUI makes it so easy to put everything on correctly and step it through. So it did take us quite a while to get up and running and everyone familiar with the process, but the actual time of putting the plastics on and getting everything run, that was really dependent on of the sample number that you were batching, the batch number you were going through, and also the type of format in the plastics you wear. But as I said, I mean, whether you're only starting off with your minimum sort of 8 samples or your entire plate, you know, with a lot more up to the 96 samples, there was such minimal increase in time of running the instrument. So the setup at the front end is a little bit longer and more a little bit more longer to put in there. However, once we had the you know, the platform up and running smoothly and the scripts all running and everything working well, it was actually extremely streamlined. But any of my students can pipe in on the actual logistics more if they have anything to say.
Yeah, so
I think
the
you may have said it all.
Oh, okay.
Go ahead. No, go ahead. Tell us, please.
The actual processing and setting everything up on the instrument, once you know what you're doing, is very, very quick. You can actually leave things on the deck to kind of make things a little bit quicker for the next setup if you know multiple people are going to be doing runs that day. And then you can load in ample tips ahead of time, so you don't have to keep reloading tips. It has locations for 3 full plates of the big tips and then the smaller tips as well. So there really is that ability to kind of keep things set up and moving forward, speeding up the process.
Perfect, thank you. Jennifer, we have another bone question for you. What was the amount of bone that you used in the method for the Nimbus Presto?
So the Nimbus Presto is using that PrepFiler BTA chemistry for bone, and so it follows the same input value of 50 milligrams for each sample type.
Gotcha, okay. Another question here. Someone's saying, we process fingernail clippings in our lab by swabbing each clipping for any DNA. Have any of you ever tried processing an entire fingernail clipping, much like the nail bed, and received successful results?
So I'll answer this one. I have not done, either swabbing of the fingernail clipping or the entire fingernail clipping personally. I follow the PrepFiler protocol for nail clippings. And so I know we do collect clippings for our staff's database, so we have them on file. I think we were able to receive some pretty successful results, but considering we were using the nail beds, they were highly more successful than just simply the nail clippings. I don't know if anyone else would agree or disagree with that.
Yeah, we have actually used fingernail clippings in the past, but not on the Nimbus Presto. We have with the BTA chemistry on the Automated Express and manually, and success was hit and miss, and of course a lot of mixtures. But that was directly soaking the fingernail clippings, not swabbing.
Perfect, thank you. Another bone question, Jennifer. How many days do you keep buried bones in EDTA solution for demineralization? Is there any special preening protocol for buried bones before demineralization?
So for this project, since we were using the PrepFiler BTA chemistry, it's a partial demin process, so there really is no pre-soaking of the buried bones using this method. However, other methods we've used have kind of been different total demin methods, so an adaptation of the Loré method where we soaked our sample in our lysis buffer, which is EDTA and then Pro-K pretty much. And we've only done that for 24-hour periods, so an overnight period. Same thing with an adaptation of the Dabney method, again, just 24 hours. I know there are other methods that do a little bit longer, but personally I have not played around with this at all.
Okay, thank you, Jennifer. Natalia, we have another question for you.
Okay.
Besides copper and brass, is there great success with other types of casing, such as nickel, steel, aluminum, anything else?
So here at Sam Houston, we haven't examined other types of ammunition. However, what we've seen in the literature is that there is greater success with non-brass and non-copper ammunition. And this really has to do with that metal ion composition. So without copper's deleterious effects, labs have been generating more STRs in their profiles after swabbing from aluminum or steel casings. That's definitely something that we can explore further and do some comparative research. However, for this project, we really wanted to stick to the most challenging type of ammunition.
Of course, why not make it the most challenging? Okay, thank you so much for that. Here's a process question. What's the BSA concentration used in the quantification step?
So for that, we did use BSA at 4.5 milligrams per milliliter. And that's not the final concentration, that is the input concentration. We used 1 microliter of that in that standard trio reaction.
Got it, thank you. Natalia, another question for you.
I'm on a roll, let's go.
You are. Since the BT mix rinse/swab protocol recovered the most DNA, would it be possible to remove some of the hands-on time by just using the solution as a swab moistening agent?
Okay, so yes, so the BT mix rinse swab protocol is extremely hands-on, does take a little bit more time than the standard swabbing, and so that's a great question. We actually did test this out here. We looked at it and we quantified how much we eliminated, how much DNA we recovered from those swabs after just doing that. And we found that no, we couldn't really eliminate that hands-on time. And I think actually the original article that the research was published in also examined the same thing. They used 4 different collection methods, and one of those happened to be just using that BT mix solution as the moistening agent. And although it did perform better than moistening with water and double swabbing that way, it did not outperform that rinse swab protocol. And so when we're talking about really low template samples like DNA from these casings, I would recommend just sticking to that standard BT mix rinse swab protocol in full to get the most DNA out of your sample.
Perfect, thank you so much. We have time for one more question. Jennifer, this one's yours. I know we were talking about the buried bones earlier. How deep were the cadaver's bones buried?
So we actually did not have record of this at that time, but based off of photographs of the recovery and at the burial, it's roughly between 1.5 to 3 feet. And this kind of goes to show that we're still seeing these challenges with buried bones, even at these more shallow depths, which I thought was kind of interesting.
Absolutely, that is. All right, well, audience, that about wraps up the time we have for Q&A today. But I'd like to thank our fantastic presenters, as well as Thermo Fisher Scientific, for sponsoring today's webinar. In 24 hours or less, this webinar will be available on demand if you'd like to watch it again or share it with your friends and colleagues. Additionally, you will receive an email with a URL to obtain CE credit documentation for your participation today. We look forward to seeing you at the next webinar. Thanks so much.